got the token to start captioning. So until then, the auto captions will work in the transcribe with the recording and the transcription of the meeting. And on behalf of ORS and NCC, I apologize.
Hello, everyone. We're going to get started shortly. We're going to give a couple minutes for people to kind of pop on, and then we'll get started.
Oh.
If you guys are just joining us, we're just going to give it a couple, another minute for people to join in, and then we'll get started. Okay, we will get started. And can you see my slides? Are we good to go?
Yes. Yep.
Awesome. Okay, so welcome everyone. We're so glad you can join us for today's NIH Office of Dietary Supplements seminar on Nutrigenomics. I'm Jasmine Hayes, and I am a health scientist administrator with ODS. In addition to organizing our seminars, I also serve as the lead for our co-funding program. Before we welcome today's speakers, let me take a moment to go over a few brief announcements. I'd like to remind you that our next ODS seminar is coming up soon. The seminar will discuss molecular mechanisms and will be held Wednesday, February 18th. If you're not already on the seminar's mailing list and you would like to be, please check the chat. We'll share an email address there. Now, a quick note about webinar logistics. To ensure good quality, everyone has been muted, but during the Q&A section, you can raise your hand and we will unmute you and you can ask your question. If you experience any technical issues, you can use the chat and we'll be happy to assist. And as a reminder, please be considerate of everyone so we can all enjoy our speakers today. Okay, so now I have the pleasure of introducing today's first speaker, Floyd Chilton, or Ski to those who know him. Ski is a professor in the School of Nutritional Sciences and Wellness and associate director of the BIO5 Institute and director for the Center for Precision Nutrition and Wellness Initiative at Arizona. Previously, he was an assistant professor at Johns Hopkins School of Medicine, a professor at Wake Forest School of Medicine and the University of Arizona, and a distinguished visiting professor of functional genomics at Tecnológico de Monterrey, and earned a PhD in biochemistry from Wake Forest University School of Medicine. The work examines how genetic and epigenetic variations interact with human diets, especially modern Western diets, to drive inflammation as well as psychiatric or developmental disorders. These precision and population-based research approaches have the capacity to provide a wide range of opportunities to enhance human wellness that include providing pathogenic mechanisms that underscore the different biologic behavior of inflammatory diseases in diverse and racially ethnic populations, discovering new biomarkers of disease aggressiveness for early diagnostic and therapeutic intervention, revealing new therapeutic intervention strategies to affect disease aggressiveness, using precision and gene-based dietary wellness and or pharmacologic interventions and creating therapeutic foods and supplements that optimize immune system and brain development for different world populations. SCI has offered more than 170 scientific publications, started 6 companies, 1 nonprofit, and holds more than 15 patents. SCI will present on nutrigenomics and vitamin D. So I am, with that, going to give you the
Okay,
into your screen. Looks good.
Okay, doke. Well, thank you so much, Jasmine. Thank you. I appreciate the opportunity to speak to this group so much. I'm just so indebted to the National Institutes of Health and ODS and NCCIH for 38 years of continuous funding, and I'm so grateful for what you've done to help me do the research that I've been able to do over my career. Today, I'll talk about ancestry-based gene-diet interactions and their role in driving health disparities, especially in inflammation, cardiovascular disease, and diabetes. Well, I'm going to go through the first few slides very quickly. Of course, diet is our leading modifiable risk factor. Diet is number one, responsible in the US for 700,000 deaths a year. Perhaps even more importantly, 14 disability life years lost to diet per year. And as bad as kind of the modern Western diet can be for certain populations, it's much worse for others. Again, just as an example of that, you can see obesity at the top, non-Hispanic white, Hispanic, non-Hispanic Black. You can also see diabetes and age-adjusted prevalence of diabetes, and you can see that certain populations are impacted much worse than others. Again, deaths from all cancers in magenta are African Americans, African ancestry populations. So certainly certain populations are impacted much worse than others. And so our work really is trying to understand the pathogenetic basis for that. We know there are other factors, but we work on the pathogenetic basis. So we work on polyunsaturated fatty acids. I was part of the original groups that discovered leukotrienes, prostaglandins, and oxylipids. And kind of the central first principle for us is nothing in biology makes sense unless in the light of evolution. And again, whether it was 300,000 years ago in Africa or 80 to 100,000 years ago or out of Africa up into Europe, across Asia, through Siberia to Beringia, and down in North America, Central America, South America, each of these populations had to evolve means for surviving. And the biggest thing they had to evolve, it was to their diets. So it's not surprising that the genetic variation for different populations are dramatically different. For PUFAs, and I'll call polyunsaturated PUFAs from now on, modern humans have undergone intense selective pressure. PUFAs are critical for innate immunity, oxylipins, energy, appetite, and mood homeostasis, endocannabinoids, brain development and function. 60% of our brain mass is fat, and 60% of neuronal membranes are the omega-3 DHA. So nutritional transitions in PUFA ingestion over the last 75 years make evolutionary discordance and maladaptation a likely outcome. So before I get started into this, a little bit about classifications. What I've shown here is the omega-3 and the omega-6 pathway. I've shown the ratio of these. Alpha-linolenic acid at the top and linoleic acid are the two major dietary PUFAs consumed. They're consumed at dramatically different ratios. 6 to 8% of energy, linoleic acid, 0.5 to 2% energy, ALA. So the ratio is about 15 to 1, that PUFA coming in. Again, these are metabolized to HUFAs, long-chain polyunsaturated fatty acids such as arachidonic acid, eicosapentaenoic acid, docosahexaenoic acid, and all of their oxylipins, which have the biology of the molecules, or all of those endocannabinoids, which have the biology of the enzymes. So what I'm going to talk about today are the steps that are necessary to create a harmful gene-diet interaction. And in particular, it's the FADS gene and polyunsaturated fatty acid interaction. Step one is there has to be a major change in exposure. And again, in 1961, still a lot of controversy around this, and I'll speak to that. But we went from 1.5% energy to 6 to 9% energy linoleic acid. So we altered the omega-6 to the omega-3 ratio from 2 to 1 to 15 to 1. And again, you can see this in this particular slide. Again, we went replacing saturated fatty acids with PUFAs, and 95% of the fatty acids in those PUFAs were the omega-6 fatty acid, linoleic acid. You can see on your left the increase in soybean oil alone from 1961. So there was a dramatic change in PUFA exposure that began in 1961 and has continued throughout the modern Western diet. So again, you're increasing the exposure, entering into this very complex pathway. You're dramatically changing the ratios of potential precursors to anti-inflammatory, anti-thrombotic molecules, oxylipins, endocannabinoids, to pro-inflammatory and pro-thrombotic. So again, do you get to arachidonic acid? Do you get to EPA, DPA, DHA? And again, it was assumed that all populations did that the same, could come through this pathway, and at most 2 to 3% energy could get to those long-chain PUFAs. Well, again, the important parts of this pathway are these oxylipins and these endocannabinoids that are precursors. So step two was, is there genetic variations in different populations? And the FADS, the fatty acid desaturase cluster, is kind of the poster child for that. And here's a couple of papers that we published, but again, this region of the genome on chromosome 11 in the 12q region controls that desaturation pathway that gets you to arachidonic acid or eicosapentaenoic acid from linoleic acid and alpha-linolenic acid. And what we showed early on in 2012, and again, have continued to show, is there's dramatic genetic variation within this pathway. And the haplotype that's produced in this part of the genome depends on ancestry. So what we showed in an evolutionary paper and Amir showed was that there was a derived haplotype that became fixed about 85,000 years ago. It emerged about 500,000 years ago and was driven to fixation in Africa. But as populations moved out of Africa, all of a sudden we began to see this ancestral haplotype, not the derived haplotype. And by the time we get to Latin America, we see a predominance of that ancestral haplotype. Now, these two haplotypes have dramatically different impacts on the metabolism of those dietary PUFAs. And again, Amir showed this initially in 2012. We had a 2012 paper in which we showed the evolution. We published a paper a few years ago. Why did it flip back to the ancestral haplotype? Well, it flipped back as populations were moving up the Bering Strait and in Beringia. In this very important science paper that came out in 2014, showed that the FADS cluster was the primary region of the genome that was responsible for cold adaptation. And in particular, flipping back to that ancestral haplotype was the major part of the genome that was responsible for surviving cold environments. And again, it has dramatic effects, huge effect sizes on size and girth. So what does that do? Again, we have dramatically different ancestral haplotypes in different populations. How does that affect metabolism? Well, this was a 2012 paper that we published, and basically in this paper, if you'll just focus on the AA, the arachidonic acid, in the blue are African Americans, in the red are European Americans, and again you see a tagging SNP which tags the haplotype. This is the SNP RS174537, and we're looking at the step that takes you from dihomogammalinic acid to arachidonic acid, and that's the rate limiting step. And basically we found several things. Number one, we found that there was dramatic frequency differences among European versus African ancestry populations, with the African ancestry populations having much higher metabolism to arachidonic acid from linoleic acid and dihomogammalinic acid. There were dramatic frequency differences. African Americans sit at about 83% of the derived haplotype. That was about 42% for European Americans. So dramatic frequency difference, still unresolved genetic differences, and there were variability within the genotypes. Well, the real question is, does this have clinical implications? Well, how important is this portion of the genome? This is a Nature Communication paper that was published about 5 years ago, and again, these were not people who were focused on PUFAs, who cared anything about lipids, but they were simply measuring chromatin interaction in EQTLs, and they showed that this region of the genome was the major morbidity region of the entire human genome. And again, perhaps it's not surprising because it is the region that's ultimately responsible for all oxylipins and endocannabinoids. So our hypothesis at the time was that if you're African ancestry, you're making much more arachidonate, and that arachidonate could potentially be driving pro-inflammatory and prothrombotic pathways. You're very familiar with the VITAL study. The VITAL study came out in 2018 and it had 25,800 participants. 5,106 were Black participants, and it found that omega-3s did not result in lower incidence of cardiovascular events or cancer. We wanted to reanalyze this once it became publicly available, so we knew that there were 5,106 African Americans in it, so we did something. We started out with optimal pair matching so that we could match the covariance. We had complete data from 3,766 African Americans. We then matched that with the other 19,000 European Americans to get 3,766 European Americans with all the covariants, and you can see the covariants here, as perfectly matched as you could get them matched. So we then used machine learning algorithms to rerun the clinical trial on this population that had been optimally matched. First of all, what you see is a dramatic reduction in myocardial infarction in the African ancestry populations on the right versus the European ancestry population on the left, where there was no difference. Perhaps most striking was we ran a weighted decision tree in myocardial infarction, and what you see is anything to the left is the most part of that's being impacted. And what you could see in the African Americans on the left was being in the omega-3 group was the leading factor that determined the risk of myocardial infarction. On the right, it did not show up in the European ancestry population. We then did logistical regression analysis with the interaction terms between omega-3 supplementation and other variables, including race. And what we showed was that there was an 87% reduction in the African Americans who were on omega-3. Again, dramatic odds ratio of 0.17. So this has led us to the recently funded trial that we're doing, funded by NIH. And as I said, we're so grateful to NIH for funding this trial. Here we're stratifying by both ancestry, and we're stratifying by genotype, and we're giving omega-3 fatty acids. Primary endpoints are fatty acid or PUFA-HUFA-derived oxylipins. How about the indigenous American ancestry population, the other side of the equation? Again, what we hypothesized was that pipe is much smaller, so you're going to get enough arachidonate, but you can't make EPA. And in fact, you're going to become EPA deficient, and that's going to impact cardiometabolic disease. And I'm kind of showing that here. If you're the derived haplotype, you're getting a lot through this step. That's the African ancestry haplotype. If you're the Emran or the indigenous American, that pipe is very, very small. Because you're starting with a 15-to-1 ratio of LA to ALA, you're going to make enough arachidonate, but you're going to become EPA deficient. And that's exactly what we showed in this paper that we published a few years back. As you can see, this is global Emran or indigenous American ancestry. This is ancestry informative markers. This is in the MESA study, and what you can see is the more indigenous American ancestry, the less omega-3s you make. And in fact, you get very, very dangerously low in those omega-3s with the indigenous American ancestry. Again, we showed the same thing with hypertriglyceridemia. There's a unique form of diabetes one sees in Latin American populations that's a very form, and again, hypertriglyceridemic, hyper-VLDL, and you saw that with Ancestry, and if you adjusted for fads, you saw most of that go away. Again, we've just published this paper, and we looked at the effect of FADS variation on omega-3 deficiency and cardiometabolic risk in Mexican Americans. And what you see here is, yes, FADS is impacting arachidonate, but you're still, with arachidonate, you're still at very high levels of arachidonate because of what you're starting with. But again, EPA, this dramatic reduction, and again, you're looking at about a 30-to-1 ratio of arachidonate to EPA in this population. Again, we looked at the impact of the FADS cluster and in tagging FADS haplotypes, and you can see dramatic effects on HOMA-IR when you're looking at the homozygous major allele versus the minor, huge effects in fasting insulin, triglycerides, and VLDL, and that's exactly what we would expect. Once again, EPA controls triglyceride biosynthesis. It controls VLDL formation. So perhaps it's not surprising if you become deficient in these, you can no longer control lipid levels coming out of the liver. We've done the same thing in Strong Heart. Strong Heart is 4,500 tribal members from Arizona, Oklahoma, North Dakota, South Dakota. And again, if you look at glucose, and in the black are FADS cluster alleles, or if you look at the impact of FADS on pre-diabetes and diabetes, you see these huge odds ratio of 2.33 for pre-diabetes and 3.25 for diabetes. Again, we still have deep concerns about the American Heart Association's PUFA recommendations. We have a paper that we've just submitted that shows if you express fatty acids as a percent of total versus concentrations of fatty acids, the relationships with biomarkers flip. And this flip, we believe, has given rise to a misinterpretation of the data as to whether these are beneficial or not. There's also this recent study that shows these odds ratios dramatically increasing as you increase linoleic acid. Again, this Sydney study that was carried out. So we're still very concerned about that. Again, I'm going to stop there. It takes a village. This has been a village of folks from my cohort in crime, Rask Matthias, who was at Hopkins and now precision medicine at NIH. But again, from institutions from Wake Forest, Maryland, Georgetown, Texas, and again, from everywhere. And again, I stand on the shoulders of giants. I want to particularly to this audience thank NIH for 38 years of continuous funding, including our current multi-center clinical trial. I could not have done this without you. ODS co-funded 10 years when I had a center with Harvard and Hopkins. So I'm very, very indebted to NIH and all that you've done for me, all the doctoral and the postdoctoral students who have been in my lab. So I will stop there and be glad to take any questions.
Thank you for that presentation. And we at ODS, we love to see kind of our involvement with funding and where that research goes. So thank you for giving that talk. Does anyone have any questions? If you raise your hand, I can unmute you and you can ask your question. James Fleet, or the presenter.
So Skeet, thanks so much. That was really interesting. I'm not going to be showing my genetics work, but I love classical genetics and this evolutionary perspective. Could you, though, give me a little more clarity on the polymorphism that you use as the kind of the lead SNP for all this work? Yeah, has there been functional characterization of what it does to FADs? Is it affecting coding, expression, or is it kind of affecting other genes in the area?
Yeah, so the SNPs that I'm showing are simply tagging a haplotype. We're still not sure what the active or the SNP within the FADS haplotype that's creating differences in the three-dimensional chromatin structure. We know that there's an enhancer that sits right in the middle of the cluster that appears to be playing a critical role. There are 2 or 3 SNPs that are thought to be functional SNPs, but again, the clarity is not. But given the size of the LD blocks, these are very good. These cover a large area, and these tagging SNPs are tagging these hypothesized functional SNPs. We just don't yet know clearly what they are.
Barbara, would you like to ask your question? You can unmute yourself if you would like to. Michael, can you help? I try to unmute her, but it's not working.
Yeah, I'm not seeing that option either.
Oh, okay. Well, I will just kind of jump in and ask a question. So is there a cost-effective way to scale up testing for these haplotypes, and how would having one affect dietary recommendations?
Well, I think it's an outstanding question. This is again, these tagging SNPs, given the size of the LD blocks, are very, very accurate at tagging which haplotype that you're sitting in. So again, there's very, very inexpensive techniques that will allow you to genotype these tagging SNPs. So it is certainly something in the future. Certainly, if our multi-center clinical trial, we want to say to NIH, let's not say omega-3s don't work anymore. Let's define the population in which they work. And I think our recently published studies of the reanalysis of the VITAL study are doing that. But I think, again, if we can have tagging SNPs that says you're an omega-3 responder because you're deficient and you're not because it's balanced already, that's going to be extraordinarily powerful. And then we can say, okay, now we know who's going to respond. Let's spend several $1,000,000 again for a VITAL type trial, but let's get a whole lot more precision in the trial.
Hey, thank you. So I know I see some other questions, but we're going to move on to our next speaker. And then at the end, if we have time, we can address those questions. And if we don't end up having enough time, I can kind of forward those questions over to the speakers and see if they can kind of answer those after the fact. So now I would like to introduce our next speaker, Dr. James Fleet. He is the Margaret McKean Love Endowed Professor in the Nutrition, Cellular, and Molecular Sciences at the University of Texas at Austin. He holds a bachelor's and a PhD from Cornell University. has previously held faculty appointments at Tufts University, University of North Carolina at Greensboro, and Purdue University. Dr. Fleet's research focused on the molecular and physiological functions of vitamin D in two areas: the control of calcium and bone metabolism and the prevention of colon cancer. His research employs the tools of molecular biology, genomics, genetics to address questions relevant to human health and disease prevention. He has published 167 primary journal articles, book chapters, and reviews, and has invited to speak on his work. His research has received funding from NIH, USDA, and the American Institute for Cancer Research. He's very involved in the research community. He's served as an editor, serving on editorial boards, standing member of NIH study sections. He has organized a workshop called the Big Data Analysis for Biomedical Researchers and served on the program committee for the Vitamin D workshop and the American Society for Nutrition and the American Society for Bone and Mineral Research. He was the recipient of the Mead Johnson Award from ASN. He was honored with a University Faculty Scholar and Distinguished Professor of Purdue, an honor awarded to just 5% of faculty. He's also a fellow of the American Society for Bone and Mineral Research. And with that, I'm going to hand it over to Dr. Fleet.
Great. Thanks so much for that introduction. I'm blushing a little bit. Can you see my slides?
Yes, you can see them.
So again, like Ski, I want to thank you for inviting me here and also giving me a chance to let people see the type of work that we've been doing in my research group. Now, before I start, I actually prefer to give credit to the people who deserve it. And on the right, I want to in particular note that a lot of the work that we're going to be talking about here is a collaboration that I have with Sylvia Christakos and Mike Verzi from a grant that we've had for now 6 years. That's from the NIDDK, but also gets some funding from the Office of Dietary Supplements. And as you'll see later, we have some work on aging in these studies that we get the mice from the NIA colony. So NIH has been very, very good to us. I want to start, though, with a fundamental reality of vitamin D. I'm going to talk to you about vitamin D and its role in calcium metabolism predominantly. And that is that vitamin D in and of itself isn't active. It gets converted first to a status indicator, the 25-hydroxy-D, which is also not active rather. And that then has to be activated into a hormonal form, the 1,25-dihydroxy vitamin D3, which I'll probably just say 125D from here on in. And the importance of this conversion is really seen very clearly in the response to a low dietary calcium intake. In this case, what you see is that you have transient reductions in serum ionized calcium that are sensed by the parathyroid gland. The parathyroid gland then produces parathyroid hormone, and it's that hormone that stimulates the induction of CYP27B1 and the conversion of the pre-hormone to the active hormone 125D. When 125D is released into the circulation, it has effects on multiple tissues. And you can kind of see this here on the right, where really what we're trying to do is protect plasma ionized calcium. And in the presence of elevated 125D, the intestine becomes more efficient at absorbing calcium from the diet. The kidney tries to retain calcium. And if those two compensatory mechanisms don't work, the bone actually then has calcium mobilized from it in a vitamin D and PTH dependent fashion. My work over the years has focused a lot on vitamin D's role in the intestine, and I'm going to focus today on the classical role that vitamin D has in calcium absorption. Our data suggests this is the single most important function that vitamin D has, at least during growth. And in this case, we have the vitamin D hormone interacting with the vitamin D receptor, a nuclear transcription factor that then regulates the expression of genes that control the uptake of calcium into the enterocyte, TRPV6, buffering and movement of calcium, calbinidine-9K, and the extrusion of calcium, PMCA1B, which is a calcium ATPase. It also induces the expression of CYP24A1, which is an enzyme whose job is to really start the degradation of the vitamin D hormone as a negative feedback group. I mention this because it sets the tone that when you're talking about vitamin D action in the intestine, you're really talking about the regulation of gene expression. Of course, work from my lab and other people demonstrate that the role of vitamin D in the intestine isn't limited to the control of calcium absorption. It can also stimulate phosphate absorption. It maintains barrier function by regulating genes related to tight junction proteins. It can regulate cell growth and differentiation, which is relevant in the distal colon for colorectal cancer prevention, and also modulates intestinal immunity, which is relevant to inflammatory bowel disease. Now, knowing that there's so many different functions that vitamin D could have, when we started the collaborative grant with Dr. Christakos and Dr. Verzi, we started to ask these questions at a more molecular level, trying to understand really things like what is different in terms of the genes that are regulated in the small intestine, the proximal intestine versus the colon, where functions are so dramatically different. And then if you kind of look at the structure of the intestine, you recognize there are stem cells at the base of these crypt structures that give rise to progenitors that then differentiate in the small intestine into the villus, and that they could have, in fact, all different responses to vitamin D because they have different biologies. And later you're going to hear me talk about things about the questions of the lifespan. And we've done this work initially in mice and also tried to extend this into human organoids, and I'll show you some of that data. Now, what you're looking at here are Venn diagrams that result when one does a global transcript profiling. And this was an RNA-seq experiment we did in mature animals, 12 to 14 weeks old, where we gave them an injection of the vitamin D hormone and 4 hours later looked for gene expression differences. And one thing you can see is there's certain genes that are regulated across all these different segments and compartments. And these are often the classic ones we've been studying for quite a long time, the CYP24A1 and the genes involved in the regulation of calcium absorption. But the same token, we were revealing a large number of genes that were specific to each segment, and it opened up our eyes and then put some weight behind some things people had seen physiologically in terms of vitamin D's function. So, for example, if you look down at the small intestine crypt, we can see that vitamin D regulates the Wnt signaling pathway, and that Wnt signaling pathway is important for stem cell maintenance. In the colon, where colon cancer is an issue, we can see the vitamin D has an anti-proliferative effect and also influences the movement of cells, the migration up from the crypt base to the tip. If you look at the small intestinal villus, where the differentiated cells are, we were also revealing some functions that we really hadn't put on vitamin D. There's a number of genes involved in the regulation of lipid metabolism, for example, that are regulated by vitamin D. Now, one of the things we were curious about is what would account for this tissue-specific regulation. And so we've used a variety of different tools, genomic tools, to do this. And I think this is one of the interesting pieces of using genomics versus studying single genes, where you start seeing larger patterns than you might with just a single gene. And so the first hypothesis was actually quite simple. We're asking the question of, was it simply the vitamin D receptor expression level across the segments? And the answer is no, that's not true. We used some chromatin immunoprecipitation to see where the vitamin D receptor was binding across the genome. And while we did see some tissue-specific VDR binding sites, it wasn't a uniform feature that accounted for all the differences we were seeing. So we looked at some other things too. Under these peaks from these ChIP-seq VDR peaks, it's actually 500 to 1,000 bases, so you can do some bioinformatics to see which transcription factor binding sites are enriched. And we started seeing that some transcription factors might be contributing to the vitamin D response in different tissues. For example, GATA4 and HNF4 are classic small intestine-enriched transcription factors, whereas CDX2 is particularly high in the colon, and you can see those transcription factor binding sites co-associating with the VDR binding peaks. And this is something we're exploring mechanistically. Of course, we're interested to see if these things are generalized in not just in the mouse model, but also in humans. And so this was work done with Noah Schroyer at Baylor College of Medicine. In his core facility, they get biopsies from patients from the small intestine and the colon, and then culture them under a variety of conditions. Under conditions with very high Wnt signaling and Wnt ligand, you get the cells to basically behave like stem cells, and you have these undifferentiated cystic organoids. If you remove the Wnt, they differentiate and become organoids that have the whole breadth of the different types of cells you'll see in either the small intestine or the colon. So treating these with 125D for a short period of time, we were able to see, again, a large number of genes being expressed, some of them which were common across all the conditions. But again, we're seeing things that are unique to either undifferentiated or differentiated states. And although it's not shown here, there are definitely differences between the small intestine and the colon. And these things functionally, when you look at the genes that are regulated, start telling you something about the biology that vitamin D is regulating. So for example, in the duodenum, we're seeing things like upregulation of integrin signaling and tight junction proteins that really are important for barrier function. Whereas in the colon, we're seeing upregulation of things like CD14 and TLR4, which are important in the response to things like lipopolysaccharides. So this would be a response to kind of insults that would stimulate the immune system. This, by the way, is a response we're only seeing in the human, not in the mouse organoids. Now, with that kind of profile landscape in mind, we actually, in the second round of our funding for our grant, decided to start looking back at an old story. And this story was originally from Alan Need and Chris Nordine down in Adelaide, Australia. And they were characterizing how aging was affecting the ability to absorb calcium from a test meal. And you can see here that basically after the age of 60, there's a progressive decline in both men and women in the efficiency of calcium absorption. Now, the original thought with that was just due to the reduction in the amount of vitamin D hormone that was in circulation. But in fact, what they later showed is that serum 125D levels don't fall with age, but they rise slightly. So this suggested there was resistance to the action of vitamin D that was accompanying aging. Now, some other studies have been done in this area. In 1973, Ireland and Fortran did a wonderful study where they fed young and old subjects either a high or low calcium diet. And in the young subjects, calcium absorption behaved exactly as one might expect, that by feeding a low calcium diet, you increased 125D levels, and you saw improved efficiency of calcium absorption in a test situation. But this was lost with the older subjects, again, suggesting something's broken in the way vitamin D is supposed to work to regulate calcium homeostasis. We've also shown this phenomenon in aged rats, and I'm going to show you that later. But this is a recurrent phenomenon across species. So in our more recent work, we're really trying to model aging using mouse models. And over to the right, what you're looking at is something from the Jackson Labs trying to relate the age of mice to the age of humans. And we're using mice that we consider young, which would be kind of late adolescence, early maturity, post-growth mature 4-month-old animals, and then old animals that are modeling kind of people 70 and above. That's a 2-year-old animal. And here I'm about to show you some stuff where we're doing some genomics, looking again at the duodenal, villus, and crypt in the small intestine, as well as gene expression regulated by vitamin D in the proximal colon and distal colon. Now, initially, we took a quick look at some genes we'd already studied, and like the classic CYP24A1 and the transporter TRPV6. And you can see very clearly that with advancing age, in these genes in particular, there's a reduced ability to induce these genes, and this could have consequence for calcium absorption and other aspects of the intestine. But that pattern isn't uniform, even for genes we know about. There's a gene that we've been studying a little bit. Dr. Christakos has a little bit more work on this. It's SLC3810, which is a manganese exporter. And while the induction is normal in the young and the mature animals, it falls off pretty dramatically at the advanced age. But of course, by using transcriptomics, we can see what's happening to all genes, not just the few genes we know. We can get out from under the lamppost and see what's the bigger picture. And so looking here at the left, this is the Venn diagram, just comparing differentially expressed genes after vitamin D treatment in young, mature, and old animals. And while, of course, there are genes that are common to them all, and this is the duodenum villus, we've also got the other tissues I mentioned, there's definitely genes that seem to be induced in one age group, but not the others. Now, this approach of using a Venn diagram actually doesn't let you see that pattern of how things change with aging. And so we use some informatic tools to see patterns. The one I particularly like is a self-organizing map. And I'm showing you 3 patterns here that we saw a number of genes follow. There's some genes that fall as you go from basically young animals into maturity. That one, for example, was the phosphate transporter NAPI2B. I mentioned to you already that CYP24 and TRPV6 have this continuous decline, and SLC 3810, the manganese transporter, falls only in advanced age. But there's so many more genes that are following those patterns, and we're trying to explore what they are and what consequence this would do. Now, this fits very much what I showed you with the calcium absorption, but there's other patterns as well that's kind of taken us aback a little bit. Just as we can see continuous declines in vitamin D regulated expression with aging, there's some genes actually whose expression increases with aging. Just to point out one, there's a protein GAD45 alpha that's involved in DNA repair. And for some reason, the expression of that gene goes up. But it kind of tells us that our simplified notion of just loss of function isn't actually what's happening with aging. It's a dramatic change in vitamin D function at the genetic level. And so what could account for this? Well, I'm going to show you a few pieces of our ongoing work to give you a sense of how we're thinking about this. The first, this is the actual data I was telling you about that we did in rats. This was when I was in Richard Wood's lab at Tufts University. And again, you can see this age-related blunting of the response to plasma 125D increases in terms of the efficiency of calcium absorption. Now, the classic action of vitamin D has to be worked through the vitamin D receptor. So later in my lab, we got vitamin D receptor knockout mouse, and we recognized that the mice that were heterozygous, HT in this, they had half as much vitamin D receptor. So this is a way for us to physiologically test the question. And our data showed very clearly that if you have half as much vitamin D receptor, the intestine was less responsive to the actions of 125D in here for calcium absorption. The problem is that a 50% lower level of VDR is not what we see in biology with aging. So back to that study from Richard Wood's lab, the maximum decline we saw from, say, 1 or 6 months to 24 months was about a 20% decline. And we actually, at the time, we didn't really love this tool. The tools in 1998 for measuring the vitamin D receptor were weak. More recently, there's much better antibodies, and we've gone to revisit this. And of course, we've got the mRNA expression now as well. And we don't have any strong evidence that VDR content is falling with aging uniformly in mice or rats in this case. And as a result, we have to look other places for a mechanism accounting for it. If you look at how a gene is regulated, and in vitamin D in particular works through a nuclear receptor, it's not just the vitamin D receptor that's involved in this, but a variety of different genes. So for example, VDR partners with the retinoid X receptor to be the transcription factor. And then it assembles co-activators and the mediator complex to unwind DNA and interact with the basal transcription units. So a large number of proteins, in addition to the VDR, are necessary. And Mike Verisi has been working on this in our collaboration using a technique called RHYME. In this case, you treat with vitamin D and you isolate the vitamin D receptor as well as any proteins that are interacting with it after you've done the treatment. And as you can see in the red there, we have a number of proteins that are enriched as a result of vitamin D treatment being associated with the vitamin D receptor. Some of those we expect. The retinoid X receptor, for example, is one of those. And some of them we hypothesized. So HNF4, in this case G, is enriched in that young animal. But when we go to the older animal, we see that the number of proteins associated with VDR decline. And so when you do a direct comparison of what you get from the VDR in the young and the old immunoprecipitation, you can see that there's a bunch of proteins that are enriched really in the young and not in the old. And we're exploring what those might be. I'll mention one of them I took an interesting look at. There's a deubiquitinase enzyme. So ubiquitination of proteins in the transcription complex can be part of the process of ending the transcriptional process. So the young animals are better at removing those ubiquitin residues, perhaps, and allowing the transcriptional response to go on longer. The last piece I want to talk to you about mechanistically is something that Dr. Christakos is just doing. In this case, we're asking questions about what is the nature of the cells in the intestine and how are they, in fact, affected by age? And then also, of course, by vitamin D treatment. And so when you do this, we did the 10x genomics CellPlex single-cell RNA-seq system after isolating epithelial cells by flow cytometry, sending our data through a Seurat pipeline. This is the type of projection we get, a UMAP projection. And one of the things you can see in blue is there's a collection of cells we can annotate to be those crypt stem cells and progenitors. And these, of course, being important for leading to the production of epithelial cells that can absorb, for example, calcium. Now, when you take a look at this in a different way, just looking at what is the distribution of these different cell identities in young and old animals, one of the things that's very interesting is that the stem and progenitor cells seem to be declining in the old group. So in this thing I just put up, the pinkish group, salmon group, is the cells from the old animals. And you can see in the stem cell group, they're just underrepresented compared to the other things. And so that would be a really interesting question. Is it simply a question that we have lost the ability to make really optimal, mature, absorptive epithelial cells? And of course, doing this work is interesting in terms of understanding the mechanism by which how things work, but we'd really like to be able to reverse them. And maybe at some point we're going to be talking about reinvigorating stem cells. But one of the simplest things we realized we could do is it's not that the old animals don't respond to vitamin D, they just don't respond as strongly. And so if you could increase the amount of 125D signaling to the cells that can absorb calcium, that would be a good thing. Now, you can't make this happen in a systemic way, because if we raise serum 125D, that is actually going not only to stimulate calcium absorption, but it's also going to stimulate bone resorption. And we think the solution to this might lie in a compound from a plant, Solanum glycophyllum. It's been well established that this plant produces a form of 125D, and that each of the hydroxyl groups can be modified by having some sort of a sugar residue on it. Here you're looking at a 1,3-diglycoside, one of the compounds that you can isolate from these plants. Now, when the sugar residues get put onto the vitamin D molecule, it can no longer interact with the vitamin D receptors. So these molecules, as they are shown here, are inert. However, you have bacteria that can cleave those and free it, and you'd end up having 125D. And those bacteria actually exist in the mouse and human colon. So bacterial glycosidases can cleave that. And now what you have is fundamentally you've targeted these molecules to the colon where 125D can act. Well, you might ask, why would I want to target the colon for calcium absorption? And it turns out that the proximal colon actually has character that allows it to absorb calcium and also to respond to a low calcium diet and 125D to improve calcium absorption efficiency. The increase isn't as large as an absolute amount, but it gets to about the same point. And one of the other interesting things is the residence time of calcium in the large intestine, in the proximal colon, is significantly longer than it is in the duodenum. So this could have a really positive impact if we could target calcium absorption in the proximal colon. So we published some pilot studies with the SG leaf to see if they could regulate some of the vitamin D regulated genes in the intestine. CYP24 and TRPC6 are the ones I'm going to show you. So we take 13-week-old mice, we feed them the SG leaf diet for 2 weeks at various doses, and then ask what happens to the expression of vitamin D regulated genes. If you look at the top panel, you see there's a really nice dose response in terms of SG leaf and expression of those vitamin D responsive genes, implying that if you have increased TRPC6, you might in fact be able to increase calcium absorption in the proximal colon. In contrast, in the duodenum, the DD, on the bottom, there is no dose response. And that means two things. One is that, as we know, the bacteria that would do this don't exist in the small intestine. And the other thing is that the 125D that's made in the colon isn't going systemic and coming back to affect the duodenum. And so we're in the middle of studies now trying to test whether or not these things we're seeing at the gene level actually play out in terms of calcium absorption and what they might do to bone. So I'll just finish by summarizing the few points I made for you. When we talk about intestinal biology, we're talking about the vitamin D hormone and its interaction with the vitamin D receptor. But vitamin D effects in the intestine are not uniform across the entire intestine. And we're trying to understand why that's the case. And we think it might be because they're relying on different partner proteins. We also recognize that aging is blunting calcium absorption, but the aging effect as it relates to vitamin D regulated gene expression does have some similarities to what we're seeing for calcium absorption. Some of those genes are following the same pattern, but a bunch of other patterns also exist, and we're trying to understand why that would be the case. Why would we have that diversity? And finally, we're hoping that we can target 125D to the colon in the future and maybe have some benefits for people for this. So I'll finish by answering any questions, but also I'll do a little bit of sales job. I'm part of a thing called the Vitamin D Workshop, and we're going to have another Vitamin D Workshop, this time in Strasbourg, France. That is funded, that conference is funded by the National Institute of Aging. So thank you so much for your time.
Thank you. That was so interesting. So we're coming up on time, but I want to get at least a couple of questions. And this one I'm actually also kind of curious about. How did you find out about the SG leaf?
Well, so my background as an undergraduate was in animal husbandry. And so I've always paid attention to what people do for nutrition in the animal science realm. And there was a group out of Iowa State, Ron Horst and Jesse Goff in particular, who had identified that this molecule could do this. It turns out it actually, in ruminants, they free it up, they eat it in the field. It's just a natural product in some fields, and they can actually get hypercalcemia. So this is kind of a manipulation of something that was actually found through animal sciences.
That's interesting. So I'm gonna ask one of the ODS staff, Adams. He asked, is there similar knowledge on the shared or unique vitamin D transcription factor binding sites in other tissues?
Well, we're working on that to some extent. As you can imagine, use of genomic tools can be somewhat expensive. So when we do our experiments, we save all the tissues, and we are looking at some of the other places, but we don't have anything to share at this moment.
Okay. So there are a lot of more questions, and I'm sure even more people have questions. So what I'm going to do is I'm going to collect all the questions and kind of send them to you all, so then we can send them back out for everyone. But thank you both for giving these talks. It was so interesting. And again, I just love to see how like ODS has helped with the research field. So thank you very much. We've all enjoyed your presentation.
Thank you, ODS. Thank you for all of your support.
Ditto. All right. Thanks again, Jasmine. I really appreciate it.
Yes, thank you. That was really interesting. I'm definitely gonna be going and giving you some questions.
I look forward to it. Have a good day. Okay.
Yep, you too. Bye.